People who publish the same paper 2 or 3 times with maybe just sending it to a different journal or changing the title and leaving the contents more or less intact have plagiarized themselves. They haven't plagiarized anybody else. So it doesn't fall into the definition of plagiarism as ORI interprets it, is forced to interpret it by the law. It doesn't mean it's right. And when we get allegations of self-plagiarism, I would send them to the university to deal with them because they're allowed to treat them more seriously. Also, the theft of other people's ideas, it's a great thing to say this is wrong, but it's nearly impossible to prove. We've had, not a lot of cases, but half a dozen cases I can remember where someone was convinced that someone published a paper on the same topic that they published on, and they probably were on a grant review committee, and they must have known about it, et cetera, et cetera. But when we had the institution look at it, it turns out that reviewer, or in fact, if that was a reviewer, in fact was doing the same research or had thought about it as shown in notebook entries prior to reviewing the other person's grant or paper. So it's everyone can have good ideas, not just one person. So I'm going to give you a couple quick examples. This is a image of some dot plots, probably RNA, in which there's 2 blocks of data that are identical, outlined in red. And you can see the little spots. There's a spot here, a spot here, the same spot here, the same spot here. Why is this important? Because what we rely on for detecting these kinds of things is often the background. Because those are like signatures that the pattern of background spots often can be very informative. The other 4 examples are examples where they're internally duplicated. This is the same blot as this. This is the same as this. See this area here? This area here? A little blur up here, a little blur up here. I don't know if you can see this well enough. You see this distinctive half moon here and here. So this has been made up of much less data than meets the eye. Falsification. Another example is the reuse of control images. These are portions of a gel that are used as what are called loading controls. So it's basically taking the same sample used to generate the experimental data and running it in parallel so that you can determine that these so-called housekeeping genes that are expressed pretty much at equal levels in all different kinds of cells, or at least the different cells being tested, it should be the same. It proves that the amount of material loaded in these lanes was the same, and therefore the difference between the experimental values is meaningful. This is Jack 2 and this is Jack 3, so they're different blots, and yet these 2 sets of loading controls are identical. This is using a gradient map tool in Photoshop, and it allows you to look at things with a little bit more carefulness because I'll explain a little bit later, but it basically takes the same image. The data is still there, it's just being looked at in a different way. And you see these shapes, like these 2 shapes here, are the same as these 2 shapes here. And the overall patterns are identical. Another example where there's been splicing to duplicate some of the data, but not all of it. These 2 sets of bands outlined in yellow represent different results with the G 5.0 concentration versus G 0.5. And this Photoshop, this tool is using a gradient map and what we call an overlay. where we take the two sets of lanes and lay them on top of each other, and this basically allows you to show that the shapes overlap completely, whereas the other two lanes do not. You see, there's all black, all red, all black, all red, or this is all red, no black. So there was more data, but I won't show it, where you can see the splice lines with different Photoshop tools. You can see the lines where that have been cut out. So that's what ORI does in terms of looking at film sometimes. And fabrication is a little bit more difficult because it means that if you make up something from whole cloth, you're not explaining how or why you did it. But we have developed a software tool that looks at numbers that have supposedly been copied from printouts from instruments. And a brief explanation is that when you have numbers that are 4, 5, 6 digits long, like these numbers here, don't bother, don't worry. Ooh, down. When you see these numbers here, it's just to show that there are a lot of them. This is a blow up of some of the numbers, and what we look at are numbers that are on the right hand of the number. So 9823, 9850 are, by some of the research that we've done, show that when people fabricate numbers, they don't do it very well. So that if you look at the distribution by chi-square analysis of the frequencies of zeros, ones, twos, threes, all the way through nines, you'll get a non-uniform distribution. And since this is biological data, sorry, wrong way here. Since this is biological data, the numbers on the left are in fact expected to be non-uniformly distributed, because you'll see a lot of 1,000s or 2,000s or 8,000s depending on what the experiment result generated. So we don't look at the leftmost digits, we only look at the rightmost digits. And so this particular experiment, there were 3 sets of these data that came on a sheet of paper. No protocols, no printouts, nothing. So the institution said that's not good enough. So 2 weeks later, the respondent generated 8 more experiments with they were all false, but they at least had printouts from the simulation counter, and those numbers were compared to these numbers with the following results. 824 question digits with no notebook records were compared with 1,021 digits from assays with records. The 2 right-hand digits were tested. The probability that the question digits were not fabricated was less than 0.00001, whereas the probability that they were not fabricated in the other set was 0.78. In other words, they were uniformly distributed. So that's one tool that we use to look for fabricated data. So other than the use of other people's language and our ideas without giving credit is what plagiarism is all about. Quoted language should be noted with quotation marks, of course, and the source should be cited. That's pretty simple, but not everyone does it. ORI has historically not made a lot of findings of plagiarism for a variety of reasons. We don't feel it's appropriate to make findings on small amounts of plagiarism, particularly in areas like materials and methods sections or introductory sections to a paper or a grant, because that language is often shared by many people. And we recognize the severity of an ORI finding misconduct on a person's career. And so plagiarism doesn't alter the truth of the scientific record very much. It's simply misrepresenting the source of where the ideas and language came from. So it's not as severe in many people's minds as falsification or fabrication of data. So if we have resource limitations, we focus on the falsification and fabrication cases. However, publishers feel very differently about that because their number one problem in their minds is plagiarism. This is actually quite striking that they consider, oops, sorry about that. I'm doing this too much. They consider plagiarism far more serious than falsification and fabrication. So here is kind of an important point. I'm not going to dwell on it because it's part of our legal fabric, not necessarily yours. But ORI must distinguish between detecting something false and stating that this represents research misconduct. In other words, making a finding. Our regulation states that it must be shown that the acts of falsification, fabrication, or plagiarism have been done with some level of deliberate attempt to deceive. So if someone is, for example, a co-author in a paper or the PI in a laboratory, and someone in that laboratory or on the paper has falsified data and the other co-authors don't know about it, that may represent a breakdown in responsibility, but that's not an issue for ORI. It's not something which meets the requirements for finding of research misconduct. And I can talk about that more if anyone wants to ask questions later. So now, with misconduct defined, we then going to touch briefly on questionable research practices, or QRPs. There are practices which do not fall under the definition of misconduct, but may violate institutional rules or violate best practices. And there have been two papers that I have read with interest. One of them is now a number of years old, by Brian Martinson and his colleagues, and the other more recently sent to me by my friend in Denmark, who was co-author on this paper by Dr. Reuter. And they just a quick summary of some of the more notable practices which a lot of people may feel are acceptable and others not acceptable. For example, not supervising your junior staff, or inadequate handling or storing of your data, or materials, your research materials. If you don't know where that plasmid is, or whether the right material is in that vial, that's a problem if they're not labeled properly, for example. Use of published ideas or phrases of other people's without referencing. Keeping inadequate notes of the research process. That's always a problem for today's scientists, where there's a lot of pressure to perform, and not keeping good records is a real problem, because how are you going to be able to prove that you did the research if you don't have good records? And ignoring basic principles of quality assurance. And there are many other areas in which some people would question a certain practice, and some of these papers give frequencies and so forth. But I'm not going to dwell on that too much. So research misconduct versus questionable research practices. Research misconduct occurs in every country. Much RM is not reported. These areas I think I've tried to cover. Questionable research practices are more common. QRPs are not research misconduct. And how do we improve science and minimize research misconduct and QRPs? Well,